Happy Friday morning. We're finally at Friday. Although the days are kind of blurry in this situation that we're currently in with working from home. I hope everyone is healthy and safe staying at home. Hopefully, you're able to get out. I recommend getting out and enjoying some of the fresh air, maybe some sunshine if we can have it again this afternoon here in Raleigh. We'll see what happens. So, for this lecture, we're going to continue talking about food safety and foodborne illness. But first, announcements. So, your PACBAC question and responses are due today by 11:59 p.m. And Quiz M10 will open today and is due Wednesday, April 1st at 11:59 p.m. If you have any questions or concerns, the two main ways to reach out and to get a response are via my email address or the Q&A forum. The Q&A forum will likely get you a faster response since more people are able to respond there. But if you have something that is of a personal nature that you need to talk to me about, that email is really the best situation there to get a response. So last time we talked about all of the different types of foodborne illness and how to stop them. We had food illness, intoxication, and toxico-infection. So with these three different mechanisms of becoming ill, really it is discussing how or classifying how the organism is going to grow or where it's going to grow. And then if it's the organism's growth itself or a toxin that it produces that makes you sick. So the source of foodborne illness can be anywhere along the food chain, whether it's the consumer or the food service section after you have the food in your hands, or the restaurant does, or from the very start at production and growth, like the romaine outbreak that we talked about. That portion was definitely the producer and the farmer that was having the contamination issue. So how do we prevent this foodborne illness from happening? Well, one of the ways we can, especially on the consumer and food service side, is to do exactly what the CDC recommends with sanitation, preventing cross-contamination, proper temperature control with refrigeration and cooking. And then on the final step, it's really managing how food recalls are occurring, whether we're reactive or preventive. And we're moving more to a preventive style of recalling before anybody ever gets sick. So we'll continue to discuss the different type of organisms that can make us sick. And so far, mostly what we've talked about is bacteria. The three different case studies that we looked at were definitely focused on bacteria, but there are other dangerous organisms out there too. Clearly, we know that in the current day and age that we are in, with the coronavirus being one of them. So, what are the different types of microorganisms that are out there, or types of things that can make us ill? Bacteria, of course, is the first one. Viruses, fungi, protozoa, and prions. And we're going to look at each of these in more detail in depth. So the first thing I want to look at with you guys is these microorganisms are very small. And so really it's very hard for us to conceive because we can't see them, how big they are. So this particular activity should allow us to see it. All right. So this animation gives us a good idea of size. And what's going to happen is as I click through the magnification, we're going to go from the size of human hair down to the rhinovirus, which is the common cold virus. Okay, so this is a pinhead needle, right? So as we move in closer, you can see the little flat head of that needle. And now we can start seeing the human hair that overlays the top of it. So we're right here in our size, okay? In our degree of magnification. We're not even to 10 times magnification with the human hair. All right, now it's really clear. So this human hair is about 2 millimeters thick. All right, as we zoom in, we start to see things on the surface. And these things that you see are dust mites. Many people have allergies related to dust mites. So that's about the size of those guys. And as we zoom into 100 times magnification, 200 micrometers, remember the hair was 2 millimeters, what we start to see is definitely what we're experiencing right now, pollen. And if it's pine pollen, it would be bigger than this, obviously, because that's what turns everything to be this nice yellow color in our environment here in Raleigh right now. But this is ragweed pollen. So we start to see that it is 200 micrometers, right? And as we zoom in, we start to see some other things. We can see the lymphocytes, which are white blood cells, red blood cells, and then Baker's yeast. So these are the three things here at 20 microns. This guy is still the pollen, right? So it is definitely large and rough on the surface, which is how it helps, how it clings to everything in sight. So we keep going. Here we go. Now we can start to see some other things. And these are the things that, that we're starting to see levels of bacteria and different types of bacteria, right? So E. coli, Staphylococcus here, different shapes and sizes, and you can start to see, oh, there's some little things in there, right? And now we're almost to 10,000 times the size initial magnification, right? So we zoom in and we can start to see the next thing on the list, and that is more of the Ebola virus. And finally, at the highest levels of magnification, we're starting to see these rhinovirus particles, which are quite small. So that just gives you an idea of scale from a human hair down to a small virus particle, how big these things are and how they operate. All right. So as we move through things, we're going to start at the bacteria level. And the first thing I wanted to go over is the difference between a prokaryote and eukaryote, which the prokaryote is more of the bacteria, and eukaryote tends to be more of yeast, that type of organism that we're going to talk about as we move through things. So the prokaryote here, as depicted, doesn't really have a nucleus that is contained within a nuclear membrane. It does have DNA material that's here in this nucleoid that is not part, or doesn't have a membrane. That's the big differential. And the eukaryote definitely has a nucleus, but it also has membrane organized organelles. So these mitochondria are present, not present here in the prokaryote. So that's some of the main differences between those two cells. The eukaryote is definitely slightly more organized, having more levels of structure versus the prokaryote. All right, so let's talk about our first group of prokaryotes, and that's those single-celled bacteria. So these are single-celled organisms, and these are further classified into gram-negative and gram-positive. And whether they're gram-negative and gram-positive is relating to if a stain binds to them or not, and the stain is called gram. So these purple cells that you see in this picture, these purple organisms, are gram-positive. They bind the gram stain, while the others are not. And these types of, some examples of these types of organisms are E. coli 0157H7, Salmonella, Campylobacter, and Listeria monocytogenes. And we've talked about different case studies that utilize some of these bacteria, Salmonella in particular. So let's think about gram-positive and gram-negative. So here on the left, you have this gram-positive bacteria cell. What you'll notice is it has this very thick membrane on the outside. This is a peptidoglycan membrane. So this is made up of carbohydrates. Right, so it surrounds the cell and absorbs lots of materials from the environment. These things are usually, pretty easy to kill. The resistance is lower because of the absorptive nature of this peptidoglycan layer on the outside. Gram-negative cells, on the other hand, have multiple layers here, right? The peptidoglycan layer is very thin on the interior of it, but it does have multiple layers that can prevent things from getting in like toxic substances that would kill the cells. So gram-negative cells are going to be more of your type of pathogenic organisms. It's not unheard of for gram-positive to be pathogenic, but the majority of foodborne pathogens are going to be gram-negative. This is our peptidoglycan chain, right? And you can see here that we definitely have something that looks more like a carbohydrate, right? We've got this 6-member chain. We've got lots of oxygens hanging off the edges of it. It has been esterified here with a nitrogen group. So we do have lots of different things going on chemically here, but overall the general structure is very much like a carbohydrate. The other thing about bacteria is there's lots of different shapes and sizes. So these circular guys here are called cocci, right? So this is very typical of that. Bacilli are going to be more of these long oblong shapes, right? And some of them are even shaped like this teardrop and have appendages, right? We can have some that are vibrio shaped. These particular ones here that look almost like a lima bean or a kama. This vibrio shape tends to be the ones for fish and shellfish that are going to make us really very, very sick. So like Kristen mentioned in her interview, these types of things are part of what we're interested in preventing with raw fish. The next level up in organization of the cell is yeast and molds, and these fall into the category of fungi. These can be single cells. They can form colonies. So yeast here are single cells, and then molds are very typical. You can see the shape of this organism, very typical of having these sort of waving branches on a stem or a base here. These are eukaryotic cells, so they do have a contained nucleus and organelles. But typically, yeast, as here, are not responsible for illness. You're not going to find those responsible for illness. They do cause spoilage sometimes. We can definitely utilize them to make fermented foods as well. But the molds can cause illness, and we'll talk a little bit more about that. These have a very wide range of growth conditions that they tolerate. Bacteria are a little bit more narrow, but yeast and mold in general some of these strains can tolerate really high acid, high levels of salt, pretty low water activities in the scope of organisms. If you'll remember, the mold has the lowest water activity that it can grow at is 0.6. Some of these are very tolerant of low water activity. None of the other organisms, bacteria or yeast, can grow at those low water activities. And again, this is not all mold, just specific types. The reasons that molds might make us sick, and we talked a little bit about which foods are okay to eat with mold and which are not, is the production of mycotoxins. So mycotoxin production is a problem. It is a problem worldwide. It's something that happens to particular products when they are too damp, and definitely in grains and legumes. You can see this peanut up here in the picture has mold growth here. So it's not the actual mold that is the issue, but it is the toxin that it produces. These toxins fall into two categories. One is mycotoxins, and one is aflatoxins. And aflatoxins tend to be more associated with, most commonly, with peanuts that we're interested in. Mycotoxins are associated oftentimes with grains, but both of these are very difficult to eliminate, partly because they're this carbohydrate structure, right? So this is a generalized structure. Not all mycotoxins or aflatoxin will have this structure, but they do have this carbohydrate structure with lots of oxygens on them. This is not a protein, you might notice. So we can't denature this with heat and change the structure and therefore inactivate it like we can with botulinum toxin. So remember, when Clostridium botulinum grows and produces the toxin, then we can just make sure we heat that sufficiently, and the toxin is no longer toxic because we've denatured it. It's a protein. Here, sorry about that. Here, that mycotoxin and aflatoxin isn't denatured by heat. So these are very difficult to eliminate once they are there. So typically what happens is once a crop is contaminated with them, we have to get rid of that particular crop. And the reason that we're so concerned with these is oftentimes things like aflatoxins, especially in peanuts, these toxins can eventually cause cancer, and they can accumulate over time and cause cancer, right? So their effects are not, you don't rebound from them necessarily in your liver. They cause liver damage and then eventually can cause cancer with that. So we have pretty tight controls as a food industry against these two types of toxins being produced. So if we think about specific toxins, ergot is a type of mold that grows on rye or wheat. So ergot's is an interesting one. You can see the structure here, lots of ring structure. It is similar to a compound called lysergic acid. And you may or may not be familiar with this. This is the active compound in LSD, the drug LSD. So if you were to eat contaminated mold or contaminated rye or wheat grains, like in the form of bread that is made from these grains, and it was contaminated with this mold growth, you would have symptoms very similar to those of being on an acid trip. Okay, so these molds cause contractions of the smooth muscles. So this happens in internal organs and veins. It happens on the exterior. It's affecting your central nervous system. And so oftentimes folks with this would look like, if they had too much of it, they would look like they were having muscle spasms or seizures. It can cause vomiting, delusions, hallucinations, feelings like you have creepy crawlies all over your skin, right? So you can see that if you don't understand what's happening, these symptoms look just wild and crazy. And there was some hypothesis that this is exactly what happened in Salem, town called Salem, Massachusetts, in 1691, excuse me. So you might remember that at this point in history, we had the Salem witch trials where numerous people were hanged because they thought they were witches. And if really what had happened, there is a hypothesis that what really happened was that some of their grains grew this mold and produced this toxin. And so the symptoms that they were seeing, they didn't understand, and they immediately associated it with witchcraft. So a theory there. But this is a problem moving into the 20th century. In the 14th to 16th centuries in Europe, more ergotism was happening. More of these grains, you can see here, these purpley blue grains, they're the ones with the ergot growth on them, right? So more of this was happening. 17th century, yellow rice disease was found in Japan, and this is from a toxic penicillium mold species. 20th century, Russia had infected grains, and even to the 1960s in England, there was a widespread problem with the death of turkeys. They were being fed peanut meal that actually had aflatoxin production in it, and this was killing off the turkeys. So not only do we need to be cautious about it with ourselves and make sure that we're not eating mold toxins, but we need to make sure that the animal species we're feeding these grains are not eating them as well. All right, so that's a little bit about bacteria, about molds and yeast, which are classified as fungi. Moving on to protozoas. So these are single-celled eukaryote species. Very often they're aquatic or foodborne, so you can get them from water, contaminated water, or eating contaminated food, and they tend to be parasitic. So the problem with protozoa is that they grow extremely rapidly, right? They multiply extremely rapidly, completing an entire life cycle in 2 hours. Typically, the type of symptoms you're going to see associated with these are gastroenteritis. Sometimes you might get muscle aches, fevers, headaches, that type of almost flu-like symptoms, but the main symptom is going to be gastroenteritis with stomach pain, diarrhea, and sometimes vomiting. The two that we classically think of are Giardia and Toxoplasma. Now, Giardia is what you would classify as traveler's diarrhea. Oftentimes, this happens when we go to places that maybe don't have the best water treatment, and we can get this particular protozoa from the water that's there, whether it's used to wash food and the food is not heated, or we're consuming water that is contaminated with it. Toxoplasma gondia is an organism that is very interesting. Sometimes these protozoa can even have mind control abilities. There's some thought there that these mind control abilities from the protozoa are quite real. Toxoplasma is gotten from undercooked meat, and especially meat that is free range, where it's coming from the animal's diet not being controlled very, very tightly. Toxoplasma only reproduced in cats. Okay, so the organism can get into another species of animal, but it cannot be reproduced except in cat, which is when we start to see these mice that love cats, right? And that they're not afraid of a cat, which is what you see right here. And so this is where that mind control ability comes in. The hypothesis is that the organism wants to reproduce. It knows it can reproduce in a cat, and so it drives the mouse to get close to the cat and not be afraid of the cat, so the cat can consume it and be infected with the toxoplasma. There are some theories that about a third of the human population does have toxoplasma. It can affect the personality. They've done studies that have looked at types of behavior associated with people that are infected with Toxoplasma gondii. They see personality difference. They see a propensity for car accidents because they tend to act very rashly. They or even some links to some schizophrenic type behavior there. So obviously, this Toxoplasma gondii is related very much so to changes in our brain and our neurology because of these different personality shifts and changes. So that's protozoa, quite interesting and quite frightening. This can happen in different organisms as well, so just keep that in mind. Cats and mice and people are not the only ones. The last is not really an organism, but it's still interesting and still frightening. So prions are very short sections of proteins. And what happens is we learned that proteins are folded in different levels of structure, primary through quaternary. And very often, that structure is related to their function in the system. And so if the protein is not folded correctly, something is going to malfunction and you're going to have a problem. So these prions get in the system and they cause improper protein folding. And specifically, the one that we're probably most familiar with is bovine spongiform encephalopathy, and that's commonly called mad cow disease. Back in the 1990s, there was a huge epidemic of this in the United Kingdom, and it basically wiped out their beef industry. So it was an epidemic that was related to 170,000 cows being infected, and they had to kill 4.4 million cows as a precaution. People can get this disease from consuming beef that is contaminated with this prion, and that's Creutzfeldt-Jakob disease. That's our human version of our mad cow disease. So it was extremely important, not just from the health of the cattle, but also the health of the human race, to make sure we can get rid of it. Now, that's difficult because all of these prion diseases that we know about are affecting the structure of the brain and neural tissue, but we cannot diagnose them prior to death, right? And they're definitely untreatable. So the way that these diseases are diagnosed typically is just from symptoms, but they cannot be confirmed until death because they have to look at the actual brain tissue to confirm this particular illness. They are untreatable. There's no known way to change the trajectory of this disease, which is typically universally fatal. So prions are pretty scary for that reason. It's very difficult to do anything about this disease once it's in play. So the way this is happening is if we consume brain or nervous tissue. So symptoms of this: rapid physical and mental decline. This happens pretty quickly. Paralysis and coma, depression, withdrawal, anxiety, paranoid delusion, all things that we can think about as being neurological diseases where you start to change your behavior quite rapidly, as well as your body function. Typically last about 14 months. And these small chains of proteins are resistant to heat. They're resistant to proteases. So we can't change their structure with heat. We can't break them down enzymatically. And we really have trouble even chemically denaturing them using a compound that would cause them to unfold. So these are very robust causes of illness here, these prions. So in 1997, the FDA banned feeding nervous tissue of ruminants and instituted culling of herds when this particular illness was found. So previous to this, there was nervous tissue of ruminants that would be fed or that would show up in the feed of some of the cattle that were there or other ruminant animals. So now that is definitely considered illegal. And the last one is viruses. These are responsible for the most foodborne illnesses in the United States. They're very small, as you may know. We've all become very familiar with viruses in the recent weeks. They're protein coat that they are basically a protein coat that contains DNA or RNA. Right? So pretty tiny. They have DNA and RNA that will get into another cell. So they invade another cell. They use that cell's machinery in order to reproduce and then infect further cells. So examples of this are norovirus, hepatitis A, and Nipah virus, which is a more emerging virus that is really found quite a bit in India. So we'll talk about a little bit of these. And of course, coronavirus falls into this category too. So let's talk a little bit about norovirus. Until recently, it was not very well understood. In fact, it was only isolated in 1968. And this virus was isolated from children in Norwalk, Ohio. So initially, it was called the Norwalk virus. It tends to be worse in the winter months. Some of these viruses are quite seasonal, like our flu. And it's easily transmitted through aerosolized vomit, water, or contaminated foods. So this can be a foodborne pathogen where when you consume it, it does begin to grow in the gut. It typically passes, we know a lot about this mechanism, from person to person very easily. You have flu-like symptoms. That's one of the symptoms that you have: body aches, fever, and the main symptom of norovirus is projectile vomiting and diarrhea that often occur simultaneously. It is not any fun at all. If you've had norovirus, you will know this. There are different strains of it. So some strains are quite virulent and have terrible symptoms, and others aren't quite as bad as symptomatically. One of the big things with this is how it's transmitted. You only need 10 virus particles to get ill. It's extremely contagious. Okay, so this is one of the big deals with norovirus. So highly contagious, difficult to eliminate from services. Bleach doesn't really, I don't think bleach puts a dent in it. I'll have to double check that and get back to you. But the big deal is that it is continued to be shed fecally for 3 to 4 days after symptoms dissipate. So somebody gets sick with norovirus. They feel terrible for about 2 or 3 days. Usually the vomiting and diarrhea is over within 12 to 24 hours. And then you continue to feel achy and terrible for another day or two, and then you feel better. So the good news is it's a short-lived virus. However, once you're well, you continue to shed the virus fecally for about 3 to 4 days. The problem with this is the person thinks, oh, I feel better, I'm fine, and you go back to work, yet you're still contagious. Okay. So you can still get lots of people sick. So helping people to understand that once you've had norovirus, you still need to treat yourself as sick 3 to 4 days after you feel better is really important to prevent spread of this illness. All right, let's talk a little bit about emerging viruses like the Nipah virus. This virus is zoonotic transmission, right? So we started with bats, and that virus moved into humans, made the leap into humans, which is where we now can see that a lot of our pandemics are coming from. It typically has an extremely high fever, encephalitis, so brain swelling, and the mortality rate with this particular virus is very, very high, 75% mortality rate. So what happened? We have fruit bats that are eating this fruit from the tree. They go up to palm dates. They are eating the palm dates or shedding virus onto the palm dates. People harvest the palm dates and then eat them, and that is where we were getting the illness from. So this Nipah virus is transferred from animals. We can also get it from pigs. If the bats infect the pigs with the fruit is contaminated, the pigs eat the fruit, they get the virus, we eat the pigs, we get the virus. So they can go through, that virus goes through that whole route. The tricky thing about this is that the mortality rate in this virus is extremely high. So they're really sort of watching that in these warmer climates, tropical climates like India and Bangladesh. I did want to share this particular graphic. You may have seen this before. I thought it was very interesting, the history of pandemics. So it gives you, not sure what happened there. It gives you the different sizes of the different types of organisms that are making folks sick. And more recently, these are definitely going to be virus-based. But you can see it gives you the timeline, when they happened, how many deaths occurred from that as well. And you can see some of these lasted several years or even almost a century, right? We start looking at yellow fever. It was in the late 1800s that it was a problem. HIV/AIDS is considered a pandemic, and it is still going on, although we've come up with lots of different treatments for that. No cures, but definite treatments. So what I wanted you to see here is this particular table. Okay, so this is the different plague and its time period here. And this is where we believe that the organism originated and the death toll coming from it. So these very early time periods, 165 to 180 AD, this particular plague was believed to be either smallpox or measles. 5 million died from it. So high death toll. Of course, here medicine was not what it is today for sure. So we didn't have very good medical care. If you got this virus, you were very, very sick and likely going to die. So mortality rate was very high with that as well. If we move through here and look at the Spanish flu, which is what the coronavirus pandemic is being compared to most closely for people, 1918 to 1919, this lasted a year. We had 40 to 50 million people die, very high mortality rate. This is a virus that came from pigs. So if you start looking at the different viruses as we move through, you can see that in most recent history, all of our pandemics are related to viruses, and the majority of them are coming from animals. Okay, so just something interesting to think about. This infographic has lots of great information. So be sure to take a look at that and investigate it more thoroughly. Let's see. Sorry, I don't know why it started over, but we'll get through it. All right. And so the last part, so those are the different types of things that can make us sick. The last part here I want us to think about is, can bacteria get sick? So we've got some very small organisms that might make us sick, but are there things that can cause bacteria to get sick? And the answer is yes. An organism called a bacteriophage, which you see here in this structure, can make bacteria sick. This is like a virus for bacteria. Now, this is interesting to think about from a number of different angles. So here's our bacteriophage particle. This is a virus that infects bacteria, and it's found everywhere. If you think about the predator-prey relationship, bacteria are found everywhere. And so, of course, bacteriophages that make them sick are found everywhere. The predator is always going to be close to the prey. And it uses the bacteria to replicate its DNA, and then it blows the cell up. And those bacteriophages are now out around the other cells and bacteria cells, and can make them take them over as well. So this is a problem if you're fermenting foods, right? Things like yogurt that use lactobacillus, lactic acid bacteria to ferment foods. If those bacteria all get sick, all of a sudden the fermentation doesn't work anymore. The other thing that we can think about doing is, so there is a problem with these, right? Bacteriophages can definitely disrupt fermented food progression and processing. But we can also use these bacteriophages against bacteria that are pathogens to us, right, that make us sick. So we could kill the pathogenic organisms by using a virus against them. That's a really interesting thought about how to use bacteriophages in our system. So when we think about bacteriophages and how they work and where we find them, really what we're thinking about is the story of fermentation evolution from something that happens on your counter or in a very small quantity in your house, and then maybe you share with your neighbor, to something that is produced industrially, like yogurt being produced industrially. So these are naturally present microorganisms, and we use them to produce fermented microorganisms. So fermentation history, let's start there. Originally, yogurt and fermented products were very small production, right? Families or neighbors, they used the bacteria that were naturally present. If we think about making yogurt, lactic acid bacteria that's used in yogurt production is present in the milk. So we don't have to inoculate anything necessarily. And that worked great for families or for one neighbor in the neighborhood or one farmer who made yogurt. Once production got a lot bigger and required more consistent production, right? So think about a yogurt manufacturer, if you bought a container of yogurt today and really liked it, and then brought it the next week and it was a completely different color and had a different texture, you might be alarmed that it was different and you might not like it as much. So these larger manufacturers, as they get bigger and bigger, they need to have very consistent product. And so the way that they were producing this product is to inoculate the milk and make sure that they were being consistent. They would take the yogurt that was already made that had live bacteria in them and put some of that in the milk that is being made into yogurt. And this is commonly called backslopping and used very often with fermentation processes. If you make sourdough bread and you have a starter in your refrigerator, you're backslopping, right? You're taking that culture and keeping it alive and using it in each batch of bread, that a little bit of it in each batch of bread that you're making. So this was utilized as the way to begin the fermentation and keep that fermentation consistent. But just like anything else, what we found is that some particular strains of organisms became dominant. And as that happened, those particular strains were the only strains being used in production, rather than having a large bank of bacteria that had a variety of genetics in them, right? So we got some genetic differences over time, but we kept them very consistent. And then all of a sudden, bacteria were very susceptible. This one particular bacteria that was dominant was very susceptible to becoming sick with bacteriophage a virus in essence. And so what we can think about then is how do we fix this problem? And this problem definitely was impacted by the development of CRISPR. And we'll talk a little bit about that next week on Monday. CRISPR was the way that we prevented bacteria from becoming sick with bacteriophage by taking a snippet of that genetic structure from the bacteriophage and inserting it into the bacteria so that they no longer would become sick with this particular bacteriophage. So we'll talk a bit more about that and how that really made a difference to the bottom line for a lot of yogurt manufacturers. So it takes all kinds of things to make fermented foods, to give us good products like yeast breads, blue cheeses, all these kind of things. But at the same time, all of these things have the potential to make us sick. And it's not based on size or any one particular thing. Almost all varieties of these things can make us sick. The only one that doesn't really is yeast. So all microorganisms, all types, and even just tiny little strands of proteins, they can cause problems for humans. So I hope you have a good weekend. Get outside, enjoy some fresh air, and I will see you again on Monday.